Hi, my name is Keith Nelson. I'm a physical chemist and a spectroscopist at MIT, and I'm just delighted to talk to you about research in a new area that is just extremely exciting to me. It's the use of strong light fields at terahertz frequencies. That's in the far infrared. 1 terahertz corresponds to 300 microns in air. And the really exciting new possibilities that are opened up by having strong fields available for nonlinear spectroscopy and control using those fields. What you're looking at in this video is you can see the waves of this field moving back and forth, in this case in a little cavity that we built, because at such long wavelengths we can actually resolve the individual wavelengths of light spatially and see the waves as they propagate. What I want to do to start is just briefly illustrate how we make these fields and then spend most of the time discussing what they are good for. We make them by using a short pulse of optical light to irradiate a nonlinear crystal. We use this crystal lithium niobate, others are available. And what this does is it actually shakes the ions around, and since that's moving charges around, a suddenly moving charge radiates. There's a burst of radiation that makes a single cycle terahertz pulse. And that can be visualized right inside the crystal. Here's an example. You're actually watching. We used a cylindrically focused line of visible light to hit this crystal and make 2 propagating waves that you can see bouncing back and forth in it. It's 3 by 6 millimeters, so it's easy to see. The way we make strong fields is not with just one line of light, but another one and another one, another one, building up a bigger and bigger field. You can watch that happen here. You'll see this image grow in strength until it's free running and then leaves. You can see it's getting stronger and stronger. Right about now, it's free running, and when it leaves a crystal, we can just use that in free space as our terahertz source. We can also make the field even stronger in local regions by using little structures like this. This is a little gold dipole antenna, just 2 strips of gold. And what you can see is that when a terahertz field arrives there, the strongest field is produced in a little micron gap in between the strips. Right there, you can see by eye that the brightest place is right there. And that's happening because the terahertz field is moving electrons away from the gap in one side toward it another. So there's a big field that's generated right there at the gap, stronger than the terahertz field that induced it in the first place. So this is used sometimes to put a sample in a location like that to have very strong field effects. And I should mention, it's possible in some cases to do spectroscopy where the sample is integrated right into a lithium niobate structure like this. In that sense, there's no need for any free space terahertz propagation at all. The terahertz field is generated right there in the crystal. It interacts with the sample and it's measured there inside the crystal. So it's a very compact, simple spectroscopic platform and definitely needs further development. The way we make strong fields is to use a bulk lithium niobate crystal. The terahertz light leaves and makes this single cycle burst, similar to what I showed you in that video. It's about 1 picosecond in duration, and that means it has a pretty broad spectrum. The Fourier transform gives us that spectrum, and you can see it extends out to about 1 terahertz in width. We can also make multi-cycle fields and tune the frequencies. There are some other ways, and there are other crystals, I should say, that go out to higher frequencies than what we've generated. Another way to do that is you can actually generate terahertz fields right in air. Femtosecond optical pulses can ionize air in a way that sends the electrons all in one direction. There's moving charge, it radiates, and that can be very short. Here's a single cycle pulse that's just 1/10 of a femtosecond. Here's the spectrum. It goes out almost to 15 terahertz. So the whole terahertz region is accessible with quite strong fields. And now, how do we use them? What's exciting that we can do with them? To understand this, it's useful to just think about how a field like this, how a light field like this interacts with molecules or materials. And it can interact in different ways. It can interact with the electrons, or it can move ions around. And so let's think about that a little bit. It can accelerate electrons or liberate them. It can ionize things. With ions, it basically is driving vibrations. It's doing IR vibrational spectroscopy, but the frequency is low. 1 terahertz is 33 wave numbers. That's lower than most molecular vibrations, but it's on resonance with lots of crystal lattice vibrations. So in that frequency range, we can certainly drive crystal lattices in many cases. We can do rotational spectroscopy. If I have a polar molecule, an electric field will tend to torque it to make it align with the field direction. But the terahertz field is in there for just an instant. It gives it sort of a kick, and then the molecule will just sit there and free rotate afterward. There's a magnetic field component. Of course, these are electromagnetic fields and pulses. And so we can use that to do magnetic resonance, but at terahertz frequencies where there's some really unique possibilities that I'll illustrate. So to show some of the possibilities that I think are really exciting, I'm just going to walk through a few examples, one or two examples of each of these, and try to illustrate the possibilities that are enabled by them. And whenever we drive any of these things fairly hard, which we can with fairly strong fields, that means we can do nonlinear spectroscopy. That is, we can make a measurable change happen to the molecules or the materials, whatever the sample is, and measure that in a nonlinear spectroscopy measurement. So let's start with the electrons. Here's an example where you're seeing visible light come out of thin strips in between gold strips that are deposited on the surface. So the gold strips are about 100 microns wide, and there's a 1 micron gap in between them. And just like that little gold structure that I showed you, in the gap, the terahertz field comes in, it's strongest right there. So all the light is coming out from the regions around the gaps. And what it's coming from are quantum dots, semiconductor quantum dots, that are just deposited all across the sample, but where the field is strongest. What it's doing is doing charge transfer. It's strong enough to yank electrons off of one dot and drive them onto the next one. Then the field is gone and the electrons jump back and there's radiative recombination and light gets emitted. So this is a really unusual kind of electroluminescence to produce this signal that you're seeing. And the really important message is that terahertz fields are strong enough to do charge transfer, and charge transfer, electron transfer, is crucially important for lots and lots of chemistry, ground state chemistry, photochemistry, photocatalysis, and so forth. Terahertz pulses can be used to induce that, to make that happen in unique ways. Here's one more example. You should be seeing lots of light, photoluminescence from quantum dots in this sample. They are being irradiated by UV light. Visible light should be coming out. But it isn't, because they're on a gold substrate. And on that substrate, what happens is electrons jump from the gold onto the quantum dots, and the charged quantum dots don't emit light. The extra charge suppresses their luminescence. But watch what happens when a terahertz field starts irradiating these quantum dots. Suddenly, you're going to see, boom, all this light comes out, because it's driven the electrons from the quantum dots back to the gold. The important point is, this happens with quantum dot blinking too, it can be controlled this way. The important point is that terahertz fields can do charge transfer, which is important in all kinds of chemistry. We've used these examples just to illustrate what it can do, but the chemical applications are widespread. One more example of what terahertz fields do with electrons: they can actually induce phase transitions, change materials, structure, crystalline structure, and electrical properties. So for example, insulated metal phase transitions can be induced this way, or in this case with single layer molybdenum ditelluride, 1 terahertz pulse actually fabricates a new crystalline structure. You can see that in the electron diffraction pattern. It's permanently changed after just 1 pulse, and so is the Raman spectrum that shows the vibrations of this new crystal lattice. Finally, of course, terahertz fields, electrical fields, will accelerate charges in free space. And there's an effort underway, it's led by Franz Kärtner in Hamburg, to try to make that happen effectively enough to reach tens of mega electron volt electron energies. If that works, it can be used to then do light scattering, Compton scattering of other terahertz fields, and shift the frequencies, their frequencies, all the way to the X-ray range. So right on a tabletop, you could have an X-ray, essentially an X-ray laser source, coherent beam of ultra-fast X-ray pulses. And that would have all sorts of just revolutionary scientific applications, but also important to other applications. There would be therapeutics, there would be biomedical applications of all kinds with coherent X-ray beam, essentially X-ray laser beams, right there on a tabletop, accessible in this way. Now let me give you just one example of terahertz fields acting on ions, not electrons. And all we're going to do is move positive ions in one direction, negative ions in the opposite direction, and that will happen because there's an electric field pointing in a particular direction. And in this case, what was done was exactly that in this crystal of strontium titanate, moving the ions far enough that they moved right into the positions where they belong in a new crystalline phase, a ferroelectric crystalline phase. So that was produced, it's transient, it's reversible. But the point is that was produced by the terahertz fields. That actually very recently, in fact, just this month, we've actually monitored that with femtosecond X-ray diffraction at a national lab facility, SLAC, near Stanford, where there's a femtosecond X-ray laser. So by X-ray diffraction, right off the crystal lattice, you can see from the crystal structure, it's oscillating. There are crystal lattice vibrations driven by our terahertz pulse. This whole system, which is miles long, there's an accelerator to produce the relativistic electrons that make this possible. That's what, in the previous example that I mentioned, we hope to see on a tabletop. Now let's talk about rotations, about molecular dipoles. So I've got my field, it's going to give a little kick and start these molecules rotating. Ordinary rotational spectroscopy can be done that way. But because I have strong fields, I'm actually going to take two pulses and do that, separated pulses. There'll be a time between them, and then I'm going to measure the responses afterward. That is two-dimensional spectroscopy. There are two time scales, the time between pulses and the time of that I measure something afterward. That's how 2D magnetic resonance, for example, works. Some of you may have seen two-dimensional nuclear magnetic resonance, 2D NMR spectra. That's used extensively in structural biology. And the reason it's used so much is because if I have, for example, the nuclear spin in a carbon-13 that's on one amino acid and a nitrogen-15 that's on another amino acid, and it turns out that the protein is folded in some way that those are close to each other. So in the protein structure, even though they're far apart in covalent bonds, they are actually near each other in real space. Then the little magnetic moment that's made by a pulse from my NMR machine that gets this magnetic moment on C13 going, it's felt by the magnetic moment on N15. So when another pulse starts the N15 magnetic moment going, the signal that's measured is a little bit different because it feels this and wobbles a little bit from this other nearby magnetic moment. That makes what's called an off-diagonal peak in a 2D spectrum. It has both frequencies, the carbon-13 and nitrogen-15 frequencies. And those off-diagonal peaks are so unique and important because they tell us about interactions between those two resonances, which in structural biology tells us about protein structure. And then different amino acids are substituted with C13 or N15, and step by step, the entire protein structure can be determined. This is a two-dimensional rotational spectrum, not a magnetic resonance spectrum, but there are two frequency axes. This is acetonitrile. It has a very strong dipole moment. So it gives a very nice 2D spectrum. This was just a demonstration of the possibility. Here is the same sort of thing in water, ordinary water vapor. And it has kind of a complicated 2D spectrum, much like 2D NMR spectra, have lots of peaks, can look very complicated. I want to focus on just a couple of peaks and mention that they are only there because my dipole from one rotating water molecule interacts with the dipole from another one. These peaks revealed intermolecular complexes between water molecules whose existence was not known before. And the amazing thing about that is water is about the most extensively studied molecule in the universe. So we might not have thought that there were many secrets left to reveal. But in fact, in the water vapor that's right around you right now, in whatever room you're in from ordinary humidity, there are complexes being formed and unformed constantly in which molecules interact, and that's revealed by these unique off-diagonal peaks in our water 2D rotational spectrum. The broader implication of that is these are interactions between two water molecules. What about water and sulfur dioxide or nitric oxide or other compounds in the atmosphere? There is a whole subfield of environmental and atmospheric chemistry waiting to be developed because we can now see complexation between molecules, which of course can have all kinds of chemical consequences that we could never see before, and we can see them in a really unique and straightforward way. Finally, in terms of one more example of what happens with terahertz fields and rotation, if I change the polarization of my terahertz field, so it starts out with a cycle like this and ends up with a cycle in the other direction, that exerts a unique torque on molecules. And if I have chiral molecules, that torque is different for the different enantiomers. So the result after it's over is that the collection of two different enantiomers are actually oriented differently in space. And that could be exploited for a unique kind of all optical separation of different enantiomers. Now, that hasn't been done experimentally. These are theoretical calculations, but that's a possibility that would just be very, very exciting. Finally, let's talk about driving spins and doing electron paramagnetic resonance, EPR. So here's an example where we did that. We just used our Terahertz field. This is a compound called hemon, and it has iron 3 in it. And transition metals have high spin states where there are sublevel splittings in the terahertz frequency range. Here's the spectrum. It's in our frequency range, and it's very simple to collect. The pulse came in, made the magnetic moment oscillate a little bit, and the Fourier transform gives us our spectrum in just the whole thing is just a few minutes on a tabletop. Hardly anyone makes these measurements. And when they do, they go to synchrotrons or use other cumbersome sources for terahertz radiation, terahertz light. In fact, this is just a simple tabletop measurement. And the fact that the duration of the signal is only a few picoseconds means that it can be done as a function of time. If I have photochemistry that's initiated, I could do the EPR measurement as a function of time after that. Here are just more of these sorts of spectra just to illustrate that this is something that can be done really quite broadly. But I want to go a little bit further and say these are just linear ordinary spectra, except not ordinary because they are at terahertz frequencies. But in fact, we can also do 2D terahertz spectroscopy. That's what's illustrated here. These are 2D terahertz spectra. These are collective spin modes of crystals, but it's the same measurement. And the point is now I can do things like look at metalloproteins that have pairs of iron or other high spin ions. Those things interact in ways that we really don't understand well because it hasn't been possible to directly measure the spin-spin interactions between them like we can with C13 and N15 when we do NMR, 2D NMR. But now it really is possible. And so there's just an entire area of molecular and biomolecular spectroscopy that is now possible that really urgently needs development with a whole bunch of exciting possibilities that should emerge. So what are the take-home messages here? I've tried to identify a few areas that I think really cry out for further development in a really concerted way. Of course, really, when these are developed, they will open up still other areas that I haven't already anticipated. But the point is, these are already areas that each of which could open up an entire subfield of work. There is no concerted funding source, either federal or foundational, to make this happen. In physics and material science, there isn't a concerted funding effort for terahertz or nonlinear terahertz spectroscopy, but there is now a recognition that these material control capabilities are interesting. So there are individual grants devoted to those sorts of areas, much less in chemistry. And in life sciences, non-existence. There is just nothing out there looking at high field or nonlinear terahertz spectroscopy, in particular like this EPR capability for biomolecules, where I think the range of applications is really just unparalleled. Let me just end with one sort of focus on EPR. And the reason I do that is because there's a very clear, well-defined need for instrument development. In those other areas, there's a clear need for methodology development. But here it's very easy to envision there should be a commercial terahertz EPR instrument that we could go out and buy. Nothing like that, of course, exists now. Just to put a point on it, here is something that's the result of a collaboration among the very few people who do terahertz EPR measurements at all. There's no nonlinear measurement, but just linear terahertz EPR spectroscopy. Let me just make a few points here. This was done at a synchrotron. There's a terahertz EPR user station at the synchrotron. Why does that exist? That exists because it answers a need that isn't already being met, but it could be met on just ordinary tabletops. The measurements were done with the samples at very low temperature at about 5 Kelvin. Why is that? These should be doable at room temperature. The signal is stronger at 5 Kelvin, but this should be something that could be done on molecules and biomolecules at room temperature. This is just their description of the fact that current EPR equipment doesn't enable this kind of work. And it's also notable that in this collaboration, there's only one US group, and there's no more than a tiny handful of groups in the US or anywhere that do this kind of measurement. One more example, I won't belabor it, but it's from the same group. I am only presenting it because it's the very rare example where proteins were looked at. I could not find a single other example from this group or any other recent measurement. So this really cries out for a truly disruptive advance. And just finally, I've mentioned that there are what I envision as real subfields waiting to be developed here. But along with that will come new scientific careers. The students, the postdocs who are involved in developing those will be in a tremendous position to start independent scientific careers and to have really exciting independent scientific careers going on to really advance these, as I mentioned, in ways that certainly neither I nor probably anyone at present already anticipates, but certainly will be out there. And there will be a tremendous range of important areas with interesting scientific and societal impacts waiting to be developed. Thanks very much.